Okay, so the title of this video is MRUB2120, MRUB2121, MRUB2122, MRUB2123, and MRUB2124 are orthologs of E. coli genes B3458, B3457, B3456, B3455, and B3454 respectively, and make up an operon that codes for the branched-chain amino acid ABC transporter in Meiothermus ruber DSM 1279. My name is Aaron, and Maddie will also be joining me in this video. So a little bit about Meiothermus ruber. It is the bacteria of interest in this study. It is of the genus Meiothermus and is a gram-negative, non-motile, red pigmented bacteria. Its name is derived from the Greek words meion, meaning lesser, thermus, meaning hot, and ruber, meaning red. It was originally isolated in Russian hot springs, but more recently has been found in paper mill pulp. It is a thermophilic species and thrives in high temperature conditions of around 35 to 70 degrees Celsius. Meiothermus ruber is a very poorly understood organism. There's not much known about the Meiothermus ruber genome. This study was conducted to hopefully learn more about some of the different biological properties that stem from M. ruber's genes. Much of our current understanding of bacteria comes from common strains such as E. coli. As a result, we used E. coli as our model organism in this study to identify orthologs in the Meiothermus genome and learn more about its biological processes. The biological process we are interested in studying in M. ruber is branched-chain amino acid transport. Branched-chain amino acids are transported into the cell via ATP-binding cassette transporters. ABC transporters are representative of the largest protein family to be identified thus far, and this is mainly because they can be located in both bacteria and eukaryotes. These transporters are comprised of 4 functional units, 2 nucleotide binding domains, and 2 transmembrane domains. Additionally, a substrate binding protein is usually present as well in the ABC transport pathway. Energy released during ATP hydrolysis is utilized by ABC transporters to shuttle substrates across the membrane. In the case of this system, the ABC transport we are studying is branched-chain amino acid transport into M. ruber cells. In this study, we analyzed suspected M. ruber orthologs of the E. coli genes B3454 through B3451 because these genes are known to comprise the ABC transporter for branched-chain amino acid transport in E. coli. As seen in the ecocyte image below, the 5 genes associated with this transporter are transcribed together as part of an operon in E. coli. These genes eventually code for the proteins LIV-F, LIV-H, LIV-K, LIV-M, and LIV-G. It is these proteins that we sought to analyze in M. ruber as well. So this slide is an overview of the different bioinformatics tools that we use throughout the course of the study. For the gene details of the genes of interest, we use the information provided in the KEGG database. The nucleotide and amino acid sequences for each gene were taken from this database and used in the other various tools to generate different data. Additionally, the DB paralog search function on KEGG was also used to identify paralogs of each gene. For cell localization data, we used TMHMM, Signal P, LipoP, Sort B, and Phobius. For the structural and functional features, BLAST, T-Coffee, WebLogo, CDD, TigerFam, PFAM, and PDB were used to confirm that the products of each gene were indeed part of the branched-chain amino acid ABC transporter pathway. To confirm the genes formed an operon, IMGM was utilized, and then finally the site-directed mutagenesis was done using any base changer.
The E. coli B3458 and MRUB2120 shared all the same outputs produced from the CDD, PFAM, TigerFam, and PDB databases. Both genes had the same CDD hit of LIV-K or COG0683 with acceptable e-values of 2.4 e to the negative 102 and 8.78 e to the negative 59 respectively. Both genes also had the same PFAM hit of periplasmic binding protein, PF13458, with e-values of 1.7 e to the negative 61 and 1.6 e to the negative 71, respectively. There were also identical hits for both genes in the TigerFam database of urea ABC URT-A, urea ABC transporter, or Tiger 03407, with e-values of 4.2 e to the negative 4 and 7.8 e to the negative 8, respectively. These hits were likely due to urea ABC transporters being similar in structure and function to our transporter. The cell localization data suggests that these genes are localized to the periplasmic space and likely code for the substrate binding protein that brings the branched chain amino acids to the transporter, so they can be shuttled across the inner membrane to the cytoplasm. MRUB2120 was found to have 2 paralogs present, while E. coli B3458 only had 1, using the DB paralog search. This makes sense given the 2 were orthologous. Lastly, these 2 genes were the only 2 that had hits in the PDB database, which were the 4-EVQ crystal structure of ABC transporter, solute binding protein in complex with 4-hydroxybenzoate with an E-value of 8.708 e to the negative 42, and the 1-USG L-Leuc binding protein with an E-value of 0. Both of these results were accurate as they indicate the functions of both genes and have E-values that make them significant hits. The E. coli B3457 and MRUB2121 shared all the same outputs produced from the CDD, PFAM, TigerFAM, and PDB databases. Both genes had the same CDD hit of LibH or COG0559 with acceptable E-values of 4.91 e to the negative 87 and 1.08 e to the negative 52. Both genes also had the same PFAM hit of binding protein domain transporter 2, PF02653, with significant E-values of 6.7 e to the negative 71 and 1 e to the negative 40. There was also an identical hit for both genes in the TigerFAM database of urea T yurt C arc, urea ABC transporter permease, Tiger 03409, with significant E-values of 2.4 e to the negative 15 and 3.1 e to the negative 31. Again, these hits were likely due to the urea ABC transporters being similar in structure and function to our transporter. The cell localization data suggests that these genes are localized or embedded in the inner plasma membrane and likely code for one of the transmembrane domains of our transporter. Lastly, these 2 genes had no hits within the PDB database, indicating that they have yet to be crystallized. The E. coli B3456 and MRUB2122 share all the same outputs produced from the CDD, PFAM, TigerFAM, and PDB databases. Both genes had the same CDD hit of LibM COG4177, with acceptable E-values of 5.12 e to the negative 53 and 6.04 e to the negative 78. Both genes also had the same PFAM hit of binding protein domain transporter 2, PF02653, with E-values of 9 e to the negative 60 and 9.5 e to the negative 42. There was also an identical hit for both genes in the TigerFAM database of urea T yurt C arc, urea ABC transporter permease, Tiger 03727, with E-values of 1 e to the negative 11 and 2.1 e to the negative 9. The cell localization data suggests that these genes are localized or embedded in the inner plasma membrane and likely code for the second of the transporter's transmembrane domain. Lastly, these 2 genes had no hits within the PDB database, indicating that they are yet to be crystallized. The E. coli B3455 and MRUB2123 share all the same outputs produced from the CDD, PFAM, TigerFAM, and PDB databases. Both genes had the same CDD hit of LibG COG0411 with acceptable E-values of 1.61 e to the negative 139 and 1.43 e to the negative 107. Both genes had the same PFAM hit of ABC transporter PF0005 with acceptable E-values of 3.1 e to the negative 32 and 2.1 e to the negative 12. There were also an identical hit for both genes in the TigerFAM database of urea trans yurt D, urea ABC transporter, Tiger 03411, with significant E-values of 3.7 e to the negative 65 and 4.4 e to the negative 71. The cell localization data suggests that these genes are localized to the cytoplasm and likely code for one of the nucleotide binding domains of the transporter. MRUB2123 was found to have 72 paralogs present, while E. coli B3455 had 70, using the DB paralog search within KEGG. This makes sense given the 2 are orthologous to one another. Similarly, these 2 had no hits for PDB. The E. coli B3454 and MRUB2124 share all the same outputs produced from the CDD, PFAM, TigerFAM, and PDB databases. Both genes had the same CDD hit of LibF COG0410 with acceptable E-values of 5.11 e to the negative 136 and 7.80 e to the negative 84. Both genes also had the same PFAM hit of ABC transporter PF0005 with significant e-values of 3.3 e to the negative 33 and 7.4 e to the negative 20. There was also an identical hit for both genes in the Tiger fam database of urea T, urea ABC transporter, Tiger 03410 with significant e-values of 4.7 e to the negative 60 and 2.5 e to the negative 32. The cell localization data suggests that both these genes are localized to the cytoplasm and likely code for the second nucleotide binding domain. Both B3454 and MREB2124 had 73 hits using the DB paralog search within K. This would make sense given the two are orthologous. Lastly, these two genes had no hits using PDB search.
Site-directed mutagenesis is a method that is used to make specific and intentional changes to the DNA sequence of a gene and any gene products. In the case of our research, we looked at the orthologs MREB2122 and E. coli B3456 and mutated their sequences using any base changer. Each had a highly conserved glycine around amino acid position 50 that was removed by deleting its 3 nucleotide codon. The figure below represents the intended mutations against the MREB2122 and the orthologous B3456 in E. coli. Functional genomics will be performed later using successfully mutated bacteria to see how the mutations mentioned will affect branched-chain amino acid transport in both M. ruber and E. coli.
Based on the various bioinformatics tools used in the analysis, it was determined that MREB2120, MREB2121, MREB2122, MREB2123, and MREB2124 are orthologous to the E. coli genes of B3458, B3457, B3456, B3455, and B3454 respectively. Each pair of orthologs was found to be a part of the same KEGG pathway, 02010, while all pairs of the orthologs were found to have identical CDD, PFAM, Tiger fam, and PDB hits with significantly low e-values. IMGM confirmed that the series of lib genes in M. ruber is transcribed as an operon, much like the E. coli orthologs. Using PFAM sequence alignments and web logo, many conserved residues were identified between the orthologs. These residues may later be used as targets for site-directed mutagenesis to see how the mutations affect branched-chain amino acid transport in both M. ruber and E. coli.